product is present in the periplasmic space. Alkali treatment is especially used for the extraction of the intracellular enzymes which are capable of tolerating high pH of say about 11 to 12 for say about 20 to 30 minutes. For the extraction of such enzymes, the cells are broken down by treating them with the alkali, and it will result into the release of the enzymes. One more is there, and it is considered as the gentle and sophisticated method, and that is the use of enzymes, where the lysozyme is the most popular enzyme used for the disruption of the cells. One more is autolysis, where the enzymes of the microbial cells themselves are used for the release of the cells. That is, this is especially used for the preparation of the yeast extract and the other products obtained from the yeast. So, these are the different methods for the disruption of the cells. Now, once the cells are broken down and the product is released into the liquid, that is when the product has come out of the cells, the next stage comes in the DSP is the product concentration. Now, here I would like to draw your attention that in some cases, some of the products, for example, the microbial enzymes, the product concentration will result into the required quality of the product, and in such products the further stages of the DSP are not required. But in other products where the further purification is required, there are different approaches as can be seen in the slide. Product concentration, it involves first the liquid extraction, that is here the separation of the product from the liquid mixture using the difference in solubility. That is, here the solvents are selected first where the products are preferentially soluble. So, preferential solubility is considered over here, and the selection of the solvent is first carried out at laboratory level. Once selecting the solvent, that solvent may then be used at the large scale industrial scale during the DSP, where the product would be more soluble, preferentially soluble in the selected solvent which is used during the DSP. And as the operation proceeds, the solvent will become more and more enriched with the product. The volume of the broth or the liquid to be processed will go on decreasing, and finally will give rise to the concentration of the product. It will result into the availability of the liquid which is highly concentrated in the product. So, liquid extraction is there. Here also it is associated or it is linked with the precipitation and evaporation, where the excess of the liquid is evaporated, which will finally result into the availability of the product in the concentrated form. Then the next is the product purification, which is the final stage of the DSP. And as I said earlier, that concentration is enough for some of the microbial products where the further stages, purification stages are not required. But in case of those products, especially those which are categorized as the high value, low volume products, which will include the bacterial vaccines, the recombinant products, that is the interferons, insulins, therapeutic proteins, antibodies, etc. So, for these types of the products which are marketed at very high value and which are generally found to be present in very small amount, very small quantity, it requires the application of the sophisticated techniques. So, as has been mentioned in the slide, the sophisticated techniques are used for the separation of these types of the high value products which are present in the broth or in the liquid in comparatively low quantity. As has been mentioned, the sophisticated techniques include varieties of the chromatographic techniques. We know that chromatographic techniques, they are very important. They play an important role in the product recovery, and varieties of the techniques are there, and they exploit different properties of the product, including the selective adsorption onto the columns, or it may be the ion exchange chromatography which exploits the exchange of the free ions, or it may be the molecular weight, or it may be the affinity, biological affinity between the biological materials like enzyme and the substrate, or the antigen and antibody, and the more advanced chromatographic techniques like the HPLC, where the high pressure liquid chromatography, where using very high pressure the separation of the product which is there in very small amount is achieved. So varieties of the techniques are there. We are not considering the details of the techniques, but at this stage you should be knowing that which are the different chromatographic techniques and what is their importance. So they are especially used in the separation recovery of these high value low volume products. Then it is also carried out using the membrane techniques, which are nothing but the sophisticated or the modern filtration techniques. Filtration, as I said earlier, that initially it is used for the separation of the solid and liquid. There the depth filters are used, whereas here when the membrane filtration, when this stage comes, the modern filters, that is it makes use of the absolute filters. You know that the absolute filters are those which are having the defined pore size, and here also the different techniques are there, like the ultrafiltration, microfiltration and the reverse osmosis. These are the filtration techniques which makes use of the absolute filters and results into the separation of the very high value low volume products which are present there in the broth. Next one is the product formulation, which is almost the last or the final step, where the crystallization. Many microbial products are marketed in the crystalline form, for example the citric acid, the organic acid or the amino acids. So various types of the chemical agents are added at this stage, which will result into the crystallization of the compounds. The excess of the liquid is removed, it is evaporated, and the product is obtained in its highly purified crystalline form. So that is the crystallization, which involves the addition of salts, solvents, polymers, or it may also involve the alteration of pH, which will result into the crystallization of the compound. And the last step is the drying, where once again, depending upon the nature of the product, whether it is heat sensitive or heat labile, the different approaches are there. In case of the stable products, relatively stable product, the residual water is removed by exposing them to the drum dryers, where they come in direct contact with the heated surfaces of the drum and the water is removed. Or it may be the spray dryers are used in case of the delicate components, labile components. The other types of the dryers are used, pneumatic dryers. Even the lyophilization is one of the methods for drying the products, the microbial cells. So after drying comes the quality assurance, which is the very or I would say the most important aspect, because before the product is introduced into the market, it is very important to check the sterility of the product, and that is the job of the QC department of the company or of the project. They would check or they would assess the product, and different methods are there, standard protocols are there, as has been mentioned in the slide. The quality assurance is tested by different methods, like sterility testing is there, as you all are already doing it in your laboratory pharmaceutical products, or it may be the bioassay, which is the only option available for the qualitative assessment of the antibiotics, or it may be the pyrogen testing, where the product is made safe, that is, it is free of the pyrogens. This is especially true for the injectables. So after quality, these we are not discussing in detail, because in the incoming series of the lecture, we are having one separate lecture where the quality assurance will be discussed in detail. But it is also, it is one of the parts of the DSP, that is why we have mentioned it over here. And the next or the final stage is the product formulation, where the packaging of the product is there, where depending upon the product, it is filled in different types of the containers. It may be the glass ampoules or the plastic containers. They are sealed and they are labeled properly. The labeling of the containers is very important, especially in case of the pharmaceutical products or the products which are to be used for the medical purpose, the date of manufacturing, the content, etc. So labeling is done, and then the product is ready for dispatch. That is, then it can be safely introduced into the market. So this, I have tried my best to give an overview of the DSP, how it is carried out in the ex situ mode of operation. The next and the equally important part is the effluent treatment, and will be covered by Bhairavi Ben. So please.
Well, thank you, Sneha-ben. It has been a very nice and lucrative discussion of downstream processes. She has given us the intricate details about the various stages that are involved in downstream processes. But you must have understood the fact that in carrying out all these downstream processes for obtaining the product in our desired form and quality and quantity, lots of waste is generated, and all that this waste that is generated is either in the liquid form or in the solid form, and this waste cannot be just directly disposed of in the environment. This waste is considered to be an environmental hazard. It is responsible for creating pollution problems, and because it creates pollution problems, you just cannot dispose it directly into the environment. And therefore the first thing that is done to the effluent is the polluting strength of the effluent is first measured, and for measuring the polluting strength we have to assess certain parameters in terms of BOD level, COD level, TDS. BOD is biochemical oxygen demand, chemical oxygen demand, total dissolved solids and suspended solids. These levels have to be first assessed. After that, if the levels are within the limits, then we can dispose that effluent without treatment. The consent limit for direct disposal of wastewater in a body of water is 20:30. That is 20 of BOD and 30 of total dissolved solids, 30 milligrams per liter and 20 milligrams per liter. If this is the limit, if you have assessed it according to that, then you can dispose it off into the environment. However, there are various disposal options available, and in these disposal options you can either, as I told you, you can either dispose the effluent directly into the environment, or you can, if the value is slightly higher than the consent limit, then you have to partially treat that wastewater before disposing it into the municipal sewerage system, not into the environment directly, but it should go to the municipal sewerage system. If the conditions are very high, then in-house treatment of the wastewater can be done, and for that, after that only you can dispose it into the municipal sewerage system. Now, the treatment options that are available, once the wastewater goes to the municipal sewerage system, after that the municipality deals with the treatment options, and the options that that municipality has is at three levels. There are biological methods, there are chemical methods, and there are physical methods. The biological methods basically are certain aerobic and anaerobic processes. Chemical methods normally include coagulation, flocculation, precipitation, and electrochemical processes whereby the COD limit is decreased. And physical methods include screening, sedimentation, or incineration. Now, as mentioned earlier, there are various stages of effluent treatment. Effluent actually is the liquid waste that is generated. Now, that liquid waste which is generated in the industry is very highly polluted, and therefore it has to undergo 4 stages of treatment. In the primary stage, normally physical methods are employed. Now, these physical methods include initially screening of the entire effluent liquid waste. In the screening, most of the large suspended solids are removed. After screening comes the use of grit chambers where small suspended particles are removed from the effluent. And then comes the stage of sedimentation where all the solid mass sediments. So basically in the primary treatment there is a separation of solid and liquid content. The solid that settles down is called primary sludge, and the liquid which is separated out, that liquid is called effluent, which then undergoes the secondary treatment. In the secondary stage of treatment, this is actually a biological process whereby we make use of aerobic and anaerobic processes to degrade the organic waste content that is present in the effluent. Now, for this biological treatment, we can make use of filtration, which you will be very much aware of the fact that there are trickling filters used where there are sand beds on which the effluent is trickled slowly, and it is allowed to trickle down the sand bed, and while it is trickling down the sand bed, microbial film develops on the surface of the sand bed, and this film is called zooglal film, and this microorganisms carry out degradation of the organic matter that is present in the liquid effluent. Besides the filtration process, even aeration is carried out, and aeration is done when aeration is used, what is basically done is that the oxygen content of the dissolved oxygen content of the effluent is increased so that the microbial flora can carry out oxidation of the liquid organic matter that is present in the liquid effluent, where the process is very commonly known as activated sludge process. There are also another biological process involved where oxidation ponds are used. This liquid effluent is taken to the ponds where that pond is exposed to air, and there will be development of algae on the surface of that pond liquid, and that algae will introduce oxygen into the liquid, and thereby whatever microbial flora is there in the effluent, that microbial flora will be able to degrade the organic matter and decrease the BOD level and COD level of the effluent. Now after that, what happens at the end of this biological treatment, majority of the BOD and total suspended solids are removed, and it has been said that about the BOD is reduced to about 25 to 30 milligrams per liter, and 85% of total suspended solids are removed from the process. Now, but still, at the end of the secondary treatment, there are lots of suspended solids still there in form of very minute particles, and along with that there is also presence of nitrates and phosphates. This nitrate, that means the nitrogen and phosphorus content of this liquid effluent is about 50% of nitrogen and 70% of phosphorus, and this proves to be dangerous. It proves to be a chemical hazard to the environment, and therefore that also has to be removed. And for removal of nitrogen and phosphorus, the next stage is the advanced stage of treatment. In that advanced stage of treatment, nitrogen can be removed biologically by the nitrification and denitrification process, and phosphorus can be removed by addition of lime, alum or ferric chloride. So this is how all the nitrogen and phosphorus content is also removed. And then comes the final treatment. In the final treatment, disinfection of the liquid effluent is done, and by using either chlorine gas or ozone gas, and after that, after the disinfection process, whereby if any pathogens are present, they will be destroyed, and then finally you have to remove, you have to carry out a post aeration process, and at the end of that you decrease the BOD level of the liquid effluent. After the treating the liquid mass of it, you have to the solid mass that has been collected. That solid mass, can I have the next slide please? That solid mass also has its